That's a hello. We are the Knights. My name is Francisco. We're going to talk today about glucagon-like peptide 1 receptor protein, or GLP-1R for short. So the GLP-1R protein assists in glucose metabolism and homeostasis. It is mainly found in cells of the pancreas and neurons in the brain. It stimulates insulin release, influences gastric motility, helps suppress plasma glucagon levels, promotes growth of intestinal epithelium. And the way that it does this is the receptor, once attached to its ligand, which in this picture here is GLP-1, it stimulates the adenylyl cyclase pathway, and in that way, it increases synthesis of insulin. And so we can see some of the other things that it does is suppresses appetite, reduces glucose production in the liver, reduces beta cell apoptosis in the pancreas, among a host of other things. Now, one of the main ligand, or what's called the endogenous ligand for the receptor, is GLP-1. But there are other what are called paralogs or similar ligands that the receptor can accept. One that is being studied extensively is exendin 4, which, depending on how long the N terminus is, has a high or a low affinity to the receptor. And depending also on that is how well it activates the receptor. So I'm going to throw it over to Justine.
Thanks, Francisco. My name is Justine Loya, and I'm going to go over some of the protein functions. It may cause satiety suppression and stimulate the glucose release in the peripheral tissue. It may also help regulate the hypothalamic pituitary axis, also known as HPA. It also affects the functions of the thyroid glands, adrenal glands, and the gonads. It also influences milk production and water balance. And I believe I'm going to pass it on to Alexis.
Mariah.
Yeah, Myra or Alexis, who's next?
Mariah is next.
Hello, Myra?
Oh, sorry, my mic was mute. My name is Maya, and I'll be going over the protein function for neurological disorders. So the GLP-1 receptor can also be used to treat neuronal injury, both acute and chronic. Stimulation of the GLP-1 receptor has been reported to show neuroprotective effects in experimental models of cerebral ischemia, traumatic brain injury, Alzheimer's disease, Parkinson's disease, Huntington's disease, amyotrophic lateral sclerosis, multiple sclerosis, and many other neurodegenerative diseases. It is also noted that the GLP-1 receptor is also expressed throughout the brain, including in the frontal cortex, hypothalamus, thalamus, hippocampus, and cerebellum. Going into more detail for one of the neurological disorders, as an example, GLP-1 receptor became a potential therapeutic target for Alzheimer's disease in the late 1900s, since diabetes, which can also use GLP-1 receptor to regulate blood glucose levels, and Alzheimer's share similar pathological features such as chronic oxidative stress and inflammatory stresses. Previous studies on Alzheimer's disease in animal models demonstrated that the liraglutide, a long-acting GLP-1 receptor agonist for Alzheimer's, could be used as a therapeutic agent for Alzheimer's, which can prevent a decline in learning and memory, increases its protein glycosylation, prevents the loss of hippocampal neurons, and synaptic loss. GLP-1 receptor can effectively improve cognitive and motor functions and significantly enhances neuronal survival. The diagram here illustrates downstream signaling pathways and cellular functions after activation of the GLP-1 receptor. Stimulation of the GLP-1 receptor triggers two majoring pathways. Both signaling pathways leads to the cellular processes of anti-apoptosis, anti-inflammation growth, and proliferation. Most downstream responses of activated GLP-1 receptor provides cellular protective functions. Now I'll be giving the slides to Alexis. Oh, wait, is there one more? So another protein function is that this protein can be found in humans. Certain residues are common in class B G protein coupled receptors across various vertebrae due to their key roles in activating the adenylyl class pathway, but GLP-1 is specific to humans.
My name is Alexis Escobedo, and I'll be speaking about protein structure. The protein structure of GLP-1R includes two unique protein chains, which is a GLP-1R with 126 amino acids. In the figure, it's the green coiled alpha helix, and glucagon, which has 31 amino acids, and it's pictured orange in the image. It is released from intestinal L cells, and it is activated by GLP-1 receptors. And the receptors are constantly using the GCG gene, which encodes common GCG, GLP-1, and GLP-2 precursors. All three peptides are encoded by different exons of this GCG gene, making it easy to duplicate. And the N-terminal of the region is unstructured, which contains all the G proteins, and the C-terminal mediates the signaling, allowing the receptors to send out signals. Next slide. The secondary structure is the C-terminus, as mentioned before, and the N-terminus. They are each on the secondary structure, and for the quaternary structure, it contains the blue chain, which is also GLP-1R, and glucagon in light green. And now I'll pass it to Jonathan.
Hi, everybody. So my name is Jonathan Diaz. I'm going to be talking about the structure and function relationship about our protein. So first of all, the GLP-1 influence shown in the diagram, it's in blue. The GLP-1R is in black, and it's an alpha helix. The red is the beta sheet. The C-terminus of the GLP-1 binds to the extracellular N-terminal domain. The N-terminus of the GLP-1 binds to the extracellular loops and transmembrane alpha helixes, TM, and then once the GLP-1 and N-terminus attached to the TM, it causes activation of the complex. And does anybody want to add a more?
Yeah, so the GLP-1 is endogenous to our body, so it's something that we create, and it was created specifically for this receptor. But there are a lot of other paralogs that can attach to the receptor to activate it. And I think, and exendin 4 is the one that I've just seen in all of the studies that we've read, or the few studies that we've looked at anyway. And the N-terminus, the attachment, so the length of the N-terminus attachment, what they're finding is, for example, the exendin 4 with the long N-terminus attachment has a high affinity for the receptor, so attaches well, but it doesn't activate it. But when they use a truncated N-terminus with an exendin 4 with a truncated N-terminus, that seems to activate it, but it has more or less, from what I understand, the same affinity as GLP-1. So and that's why they're using that in medication, and why it gives some of the great benefits that I believe Alexis was talking about with the neurological diseases. And then finally, here are our references. If anybody wants a copy, we can always send them a copy of the references. Anybody else want to add anything? Nope. All right, I'm going to go ahead and stop recording.